All right, so you can see the slide, right?
Yes, sir.
Okay, all right. Yeah, so we'll talk about synapses today. And so what we spoke until the last class was what are the kind of distinct components of neurons. So you had cell body, and then you had the axons and the dendrites, and the axons made synapses on the dendrites of the next cells or the soma of the next cells, or even sometimes the axon of the next cells, and as well as the dendrites received information from the presynaptic cells. Now the question is, how are they communicating with each other, and what are the modes of communication, and how do you study these modes of communication, which is the synapse, or which is the connection, basically the interface between two cells. And also the what we up with is how, so basically how, so now there are different components of synapse. There are different molecules expressed. There are cell organelles that are kind of permitting the interaction between two cells through the synapse, and how we're studying them, and also how we are studying the molecules that are involved. So the first thing, so there are primarily two kinds of synapse, and in mammals, so we mostly have chemical synapses, and also there are some electrical synapses. And most of the synapses are in our body, in our nervous system, are chemical synapses, but there are also electrical synapses. So the first is we'll go over very briefly electrical synapses. And so the idea of electrical synapse is that there is something called a gap junction, which is not a so like a chemical synapse, which I'll come to later. And so the gap junctions are basically this protein. There is an interface. There is a 3 or 4 nanometer gap between two cells, and there are receptors that are interconnecting these two cells. So there are receptors that are expressed on this side of the membrane and other side of the membrane called connexins, and the daily discharge can just pass through it. So when you are recording from one cells, and so there are two cells, and so this is how the structure looks like under electron microscope. And as we spoke about before, the different microscopy techniques has different resolution. The electron microscopy has the most resolution, so you can actually see at that level, which is like few microns long, which you won't be able to see under a light microscope. And even the what you know now as the super resolution microscope, they can't resolve to this extent. So they're still limited to 10 to 20 nanometers, which is a way higher resolution than a light microscope, but still it won't be able to achieve the resolution of electron microscope. And so this is the structure of it, and then you can record, which is record means here that you're basically recording the voltage, the relative voltage between the two cells. And so this can be neurons in, this can be a neurons in hydra or any invertebrates or a mammalian system. And this is not an actual recording, and what they try to show is when there is a action potential, which the change in voltage, the communication that happens through a gap junction is called a PSP, or postsynaptic potential, which is the voltage from so the action potential is basically coming down this axon, and there's a voltage change. So if there's a signal sent from this cell, the presynaptic cell, to the postsynaptic cell, there should be a voltage change here. And if the communication is through a gap junction or electrical synapse, it's called the electrical PSP, or postsynaptic potential. And the voltage change, if you see the recordings for the chemical synapses which come later, they would be much more dramatic, and the changes are basically much smaller. So, but why do we need two kind of synapses, right? So that's another question. Why can't we have chemical synapses, just chemical synapses? And so there are various hypotheses for this, and one of the hypotheses comes from the recordings that did between these two, which we haven't done neuroanatomy yet, but when we come to that, we'll talk about this again. So there's this region called cerebellum, which is basically our back of our brain, and it's involved primarily in motor coordination. And there is this structure called olivary nucleus, and the olivary nucleus is in the brainstem, and it sends this long projections to the cerebellum. Anyway, that's not the most important part, but the most important part is what the scientists found that so the oligonucleus is basically sending a bunch of axons to the cerebellum, and what they found that when the gap junctions are there, the cells, when they're recording from the cells, they show this oscillation or this slight change in voltages, like, and if there are two cells next to each other which are presumably connected, they're all they're firing in a very coordinated pattern. So there are these oscillations and actual action potentials. So they're firing in a very coordinated way. And if you can imagine our nervous system, there are a lot of things that we do, a coordination between um, two hands or two fingers, even two next to each other. It's or when you are just speaking, the coordination between your tongue and the rest of your say the rest of your face, it's extremely important. And when a brain structure is controlling something like that, then it needs to have kind of coordinate, like the different neurons that are controlling different parts of the body. Say you are talking and using both of your hands, and the left hand is coordinated to the right hand. So similarly, the neurons has to be kind of tuned to each other so that your actions are perfectly coordinated or they go as you want. So that's why the coordination is important. And what they found was that with when the gap junctions are there, so they did it by knocking out a protein that is important for the gap junctions. And when the gap junctions are there, the oscillations are there, and then there are kind of coordinated action potentials. But when they got rid of all the gap junctions, so they found like both the neurons still can fire action potentials and have the oscillations, but they are not coordinated with each other. So they think that the chemical synapses are, yes, the primary mode of communication between two neurons, but you also need gap junctions to some of the kind of behaviors that needs neuronal uh, circuits to work in tandem with each other. And so this is an experiment that, so it's not uh, from the same system, so that they're recording from cerebellum. And so this, what they're recording is, so they're recording, so this is a behavior, the mouse basically, they're giving sugar water, and the mouse are licking the spout. So the experiment is that they basically, it's a Pavlovian conditioning paradigm, which is like very simple, where you give a mouse a tone and you give a sugar reward. So when you give the tone, and so you train the mice on it, so you give the tone and the mouse gets a sugar reward. So the mouse, basically, you don't give the mouse any water for a couple of, like, for a few hours. The mouse is really thirsty, and then the mouse kind of makes a connection between the tone and the sugar reward it gets every time there's a tone, right? So what happens is, then you give the tone and you don't give, even if you don't give a sugar reward, the mouse will lick the spout. So mouse kind of learns by itself that, that tone means the reward is going to come of sugar water. So that's the experiment they're doing. So there's a number of the movements, the tongue movements. This is the zero time point is when the tone comes on, and there's a latency, and the mouse basically starts licking the spout, and they're counting the number of tongue movements. They're measuring by the number of touches the tongue makes on the water bottle. So, and then at the same time, they're recording from the cerebellum neurons that we were talking about before. So, like, what neurons they're recording, those are called Purkinje neurons. That's not the most important thing. But if you remember what I said in the last slide, that is in the last slide, where neurons need to kind of work in tandem with each other, they need to coordinate with each other. So what they found was that so in the cerebellum and the Purkinje cells, when they are recording from one, both neurons that are connected to each other, they that are connected to each other, they kind of fire in a coordinated way. And this precedes the tuck movement, so that's kind of implies that the brain is basically computing, okay, I need to do this, and what do I need to do in sequence. And so these neurons can predict the tuck movements. So, and then we go to the major kind of synapses that are called the chemical synapses. And the we went over the like basic structure of neurons, and you now know, by now you know, then you have axon terminal, and it's making a synapse on a postsynaptic dendrite, or an axon, or a cell body, which have different names. And the basic structure is basically you have this ending, which is loaded with mitochondria, synaptic vesicles, which carry the neurotransmitters. And also there are kind of different kind of neuromodulators that are secreted. And then on the postsynaptic side, you have different proteins that are receiving and kind of transmitting the information that are carried by neurotransmitter in the synaptic vesicles, and then transmitting and continuing the action potential in the postsynaptic cells. And also there are these regions called active zones. These are the region where all the synaptic vesicles are coming and fusing there and transmitting the information. So how does a chemical synapse look like? So this is that is a graphical representation, and then this is how it really looks like. This is the electron microscopy again. So electron microscopy, as I said before, that can resolve up to a few nanometers. And I also talked about before that synapses are the space which is around 20 to 30, so between 10 to 30 nanometers. And you can still see so there's a central synapse, and you can still see the gap. And so this gap is basically filled with something called extracellular matrix, which is basically proteins that are providing support and also providing the necessary condition for the neurotransmission to happen. And also the neurotransmitter, so you will come to that later. So say, for example, that there is something called acetylcholinesterase. So the neurotransmitter, when it crosses the membrane, synaptic vesicle fuse here, this place called active zones, and then release the neurotransmitter. If the neurotransmitter hangs on too long, then that's a problem, because then it will, go on, because the neurotransmitter needs to, after once action potential is passed or the transmission happens, it needs to stop. It needs to stop the information there, and so the neurotransmitter needs to be cleared up. And so that's why the extracellular matrix is extremely important, and actually has a lot of proteins to make sure that this is transmitted and the information comes and stops as required. Um, yeah, and then there's something called, we come to this later, something called dense core vesicles. So which carry something called neuropeptides. So there are synaptic vesicles and dense core vesicles. So the synaptic vesicles carry some neurotransmitter like glutamate or GABA, and dense core vesicle will carry neuromodulators or neuropeptides, such as substance P, which will come again later. And the synapses can be of various kinds. So the axon can make a synapse on the dendrites, and it's called axodendritic, or the soma was called axosomatic, or to another axon, so axonic. So this kind of synapses are mostly in the central nervous system, which means the brain, but also our periphery has synapses, such as neuromuscular junction. That is the connection between the motor neurons and the muscles. So, but the dendrites is a property of mostly brain neurons, or central nervous system neurons. Dendrites are really abundant in cortical neurons, so dendrites are not abundant on every kind of neuron. So even though we're describing that, we are generally saying these things because we're talking about brain neurons or brain synapses and brain connections. It's a little bit different when you go to the periphery, or you go to the less evolved animals. And how do you visualize, how do you do even, these are all drawings, so how do you even look at them, how do you see the dendrites? Now electron microscopy, as you saw, if I don't tell you what they are, it's very hard to guess what they are. So you kind of have to believe people. They don't look like, because the resolution is so high, the things are so small, that it's kind of hard to imagine a zoomed out picture or thinking this is actually a part of drawing like this. So how do you even know that? So that's why fluorescent techniques are very useful. So this is um, so basically the neurons filled with dye. So they put fluorescence dye and fill the neurons, different kinds of neurons with it. And since they are filling the dye in one neuron, so of course this space is all full of hundreds and thousands of neurons. But the fact that only one neuron is lit up, you're just seeing that and not seeing the other. So you can kind of, using light microscopy, you can image them. And if you see here that the zoomed areas are where the dendrites are, and they're focusing on it, and the dendrites have this beaded structure the spine, the dendritic spines. The spines are the beaded structure on the dendrites, and this is full of. So one idea is that these are the where plasticity or learning happens, which means that these are very dynamic things that they come on and go off, and they are talking. They're very dynamic in the sense that a lot of people believe that this is where memories are, at a very subcellular level, a molecular level. That's where memories are stored. And when you don't have dendrites or less dendrites, so a lot of disease models, or even in patients like Alzheimer's or neurodegeneration, people do think that dendrites are the first to go off. And if you can protect the neurons, you can kind of make patients better. So there's a lot of disease implication, and it's also fascinating structures, because these tiny structures, they're very dynamic. These are loaded with different kind of neurotransmitter receptors, neuropeptide receptors. The receptors can differ from the region of the brain, from the functions they do. Over age, they can reduce, they can be really high. The number of dendrites can be really high when you're young. As you get older, it kind of reduces. So it means during the growth, you need a lot of more processing. So there are a lot of physiological events in our lives that can be attributed to more or less dendrites. So this was a central nervous system kind of overview of synapses and synaptic structures. And as I said, that synapses are not only in the central nervous system, but also in the peripheral nervous system, such as the connection between the motor neurons and the connection between the motor neurons and the muscles, which is called the neuromuscular junction. And it's quite fascinating. So why neuromuscular junction is important? Because a lot of information that we know now about synapses came actually from neuromuscular junction, because it's a much bigger synapse. It's hundreds of times bigger than a brain synapse. So now we have the tools to understand brain synapse pretty well, but back even a few years back, it was pretty hard to study. So a lot of things, a lot of discoveries, that's why it was made using neuromuscular junction as a model. So, but the basic structure is same, and so it's historically very important. And a lot of disorders, such as myasthenic syndromes, where you have muscle wasting, or even after injury, or in aging. So when people get older, one of the major things happens that we lose muscle mass, and one of the ideas is that it's because of loss of neuromuscular junction. So you have less neuromuscular junction, or your muscles are maintained. So if you can there's a lot of interest that if you can make neuromuscular junctions function better, you might be able to restore or slow down the loss of muscles due to aging or healthy aging. That's the kind of the aging that happens without without any disease or anything like that, just getting old. And if you can, idea is if you can restore some of the neuromuscular junction or protect the neuromuscular junction, then the muscle loss will happen much slower. And it did indeed a lot of experiments now that are done in mice and rats, and which show that some certain proteins that can be knocked out and kind of uh, or overexpressed to make neuromuscular junction retain better, and that also reduces muscle loss. So, as I said, is quite important, and they look very different in different kind of animals. They have different neurotransmitters. They have different transmitter receptors, and this is like a few cell animal, very simple animal, which is a tardigrade to amphibians, reptiles, birds, and mammals. So mammals kind of, as you see, they kind of have a similar structure. Uh, but as I said, they have different receptor and different ways to work. And it's quite fascinating, like, how the structures evolve and why they evolve and why do we have, I mean, these are all, even now, this has been studied for years. Even now, if even from an evolutionary perspective, like, think about it, why these varieties of receptors, harmful receptors. So we don't know that answer. And one fascinating was that the lot of things that we know about the molecules of synapse came from this studying neuromuscular junction in electric ray fish. And this is fascinating because they have this head in the front part of the body. They have something called electric organ, which is basically a very it's a muscular structure and it's full of energy. So they can give you electric shocks just because they have so much, so much neuromuscular junction concentrated there, so much potential is built upon it that they can discharge the electrical charge whenever they want, and they can decapacitate or attack the fish, human. Uh, since this is like thousands and millions, hundreds of thousands and millions of neuromuscular junction in a very small, intensely organ, if you want to study what are the molecules at synapses or neuromuscular junction, it's a better thing would be to just isolate that and study. And one of the reasons that I'm kind of harping on neuromuscular junction so much, so I did my PhD on neuromuscular junction. And so, but anyway, the point is that I just wanted to show you some experiments where, so this is from mice, and this one structure is one synapse. So every muscle fiber, if every so all of our muscles have hundreds and thousands of fibers each, and each fiber has a synapse on it. And this synapse is basically stable and it stays on there for whole life. You don't lose it unless you lose that muscle fiber. So these are very stable structures, and they just look like this, like since you are 10 years, 10, say a year old to say you are 60 or 70 years old. So it's extremely stable structure. It doesn't change much. Uh, and so this is called a pretzel-like structure. So this is like a bakery pretzel, and the red is bungarotoxin, which is basically a toxin that binds to the receptor, and then the green is the nerve. Blue is the Schwann glial cells we talked about before. So you can even in a simple structure like this, you can see all the three components. That is the muscle side of the synapse, the nerve side, the glial side, and you can see them together. Yeah, and then what are the different kind of neurotransmitters? Sorry, one second. Yeah. So what are the different kind of neurotransmitters? And then so there are different kind of flavors if you might say. So there are amino acids, there are GABA, glutamate, and glycine. There are amines. So it depends on their protein peptide properties. And there are amines, which is acetylcholine, epinephrine, histamine, and then you have peptides. And Yeah, so and peptides, and so these are basically these molecules are uh, getting synthesized in the cell body in the as we talked about it comes from the mRNA, it's getting packed through it's getting translated through the ribosomes, and then it's packed into neurotransmitter vesicles, and then eventually in the vesicles, and they go to the axon terminal. And in this also there is some processing, so they need a precursor molecule that is basically the protein that is coming here, synthesized and coming here. Then there is a enzyme that makes it a neurotransmitter, and then you have a transporter protein that is packing into synaptic vesicles. So if you just read the textbook a bit about it, that will tell you that how this is a very simple procedure that that is converting the precursor molecules that are coming from the Golgi bodies in the and then making it into neurotransmitter packed into synaptic vesicles. And these synaptic vesicles are getting released into the extracellular membrane. And as we talked about before, that not this we didn't talk about before. So the idea is that the synaptic vesicles has specific sites that they can release, so they can't just release anywhere. And this happens because of calcium intake. And how do the synaptic vesicles decide where to fuse, and why is it important for a place? So there are receptors just opposite to it here. So when these synapses are formed, or when the nervous system is developing, one neuron is pairing with the other neuron. So when they're pairing, there is signals that are going from this side to that side, to that side to this side. So this side will say this side to express the receptors here and kind of concentrate it, and this side the signals will say to make the active zone here. So active zone is basically where all the synaptic vesicles are fusing and releasing the neurotransmitters. And also the active zones are happening because of expression of specific proteins. And if you look at the electron microscopy, if you just Google active zones or synapses, you can see the specific kind of dark structures that are. So I'll show you here. So here are the active zones. So here are all the, similar to the cartoon, here all the synaptic vesicles are coming and fusing. And on the postsynaptic side also you can see dense structure because there is a lot of proteins that are expressed here. So, yeah. So, and it's releasing by exocytosis that you might study in other cell biology classes. And and when the neurotransmitter transmission happens, and as I said, so in neuromuscular junction, this acetylcholine, and once it's released, there are proteins here, which is called esterase. In case of acetylcholine, is clearing up it really fast so that this cycle can happen again and again. Yeah, and then so how do you do the experiments, and how do you record them? How do you know that? So this is a very this is when you're looking at it in a microscopy or in electron microscopy, the cell is already dead. So how do you even know what is happening in dynamic fashion? And one way to do is to do electrophysiology. So when you do what do you mean by, again, we talked about electrophysiology before. You're basically taking an electrode or a glass electrode, and you're filling up with potassium chloride, and you're poking cells. So what is yeah. So the when they're recording from the postsynaptic cells, uh, so depending on the kind of current, so if it's excitatory so that as I said, there are different kind of neurotransmitters, right? And so the neurotransmitters, one can be kind of divided into by is there amino acids or amines or neuropeptides, or this is the kind of proteins they are. or they can be something like excitatory or inhibitory. And what do I mean by excitatory and inhibitory? So excitatory means that one neuron is exciting the other, so activating the other. So this actually continually saying, okay, you fire, so this neuron fires, postsynaptic neurons fires together. Or it can be inhibitory, which means that one neuron is asking, it's silencing the other neurons. So if you in a very simplistic term, that if you want to stop doing something, or your brain is telling, and one neuron is telling the other neuron, just say like a good example would be that even in your say in your spinal cord or in your brain, so you have a feeding neuron that when you're hungry, the neuron fires, and or the firing of the neurons is making you hungry, and then you are eating or you want to eat. But there needs to be inhibition to the countering that effect, that once you're full and your body is basically sending the signal to your brain saying that your stomach is full, you don't need to eat anymore. So the same neurons need to be inhibited. And if the inhibition is gone, you're going to eat more. Or if the activation is gone, so of course you're not going to eat. So the thing is that should be balanced, and then that's why you have both excitatory and inhibitory synapses. And so when they're recording from the postsynaptic cells, and the synaptic neurotransmitter, the excitatory neurotransmitters such as glutamate is coming and binding the receptors on the postsynaptic cells, what is happening is you see a change in potential that will make the, eventually that will kind of reflect that. So this is the, sorry. So this is the, what happens is this is what is at the resting state. So when you are seeing the action potential going, so that kind of that's an event is happening or a communication happening between across the synapse. But the excitatory postsynaptic potential is the resting potential. By that, so if you're seeing this potential here, if the potential changes, there's a synaptic release. And how do you know there's excitatory synapse or inhibitory synapse? So depending on the receptors or the channels, the neurotransmitter receptors that are expressed, you can use a drug and block it. And if this doesn't change or this changes, if you block it and you don't see this, or it reduces, that that you're looking at a excitatory synapse or an inhibitory synapse. So if it's not very clear, this will make a little more. So it's same way when you have inhibitory synapse. So you have sodium channels here in the postsynaptic site, and that's what decides it's excitatory synapse or not, or you are recording EPFC or excitatory postsynaptic potential or not. And depending if the chloride channels, that will it will look like this, the potential, and then that this is a inhibitory synapse. So depending on what kind of signals from same recording preparation, everything is similar. You're just poking that you're not doing anything. You're not. You're activating the presynaptic neuron, but the kind of potential alteration you see decides if it's excitatory synapse or inhibitory synapse. And also, there is not just so the receptors that I were talking about. So those are mostly channels, but not all neurotransmitters happen through that, and there are other neurotransmission mechanisms which happen through GPCR, so G protein coupled receptors. And so what do they do is, so this is a much slower system, and the GPCRs bind to their kind of component supporting proteins. So GPCR are G protein coupled receptors, and so the G proteins are basically these small intercellular proteins that are they're always close and kind of interacting with the actual the receptor, which is a transmembrane receptor here, the GPCR, and all of them together is transmitting a signal and kind of modifying the action, modifying the working of the ion channel. Or so a neuro, that's what they call neuromodulator. So a neuromodulator can modify the activity of a channel here, right? So say so then a neuropeptide or neuromodulatory molecule can kind of work together with a neurotransmitter and modify how the neurotransmission happens. So it can slow it down or it make it faster, it can potentiate it, which means that it's now much easier to fire. So the EPSP or IPSP, you will see, it will see much faster changes or much bigger changes. Yeah, and then so, and also the something I would like to stress on are neuropeptides. So the kind of neurotransmission we're talking about, the kind of neurotransmission we are talking about are uh, that happens to the kind that happens to the amino acid, the amines, and but you have the neuropeptides, and so there is the peptidogic neurotransmission. And one of the so we still don't understand how neuropeptides work and how neuropeptides are processing information and transmitting information, and they do through this, which looks much darker than the synaptic vesicles and are called the dense core vesicles. And even though the neuropeptides were discovered in 1930s, almost similar time as neurotransmitter, our understanding of that is much less. So there's a lot of room to be, even though a lot of investigation has been done for neurotransmitters, a lot of room to understand how neuropeptides work and how these dense core vesicles work and how they fuse, which we, as I said, we know a lot about neurotransmitters, but not the peptides, the modulators. And how do we study that? And so, what this is a kind of a summary of what happens in real life. So you have excitatory or inhibitory neurotransmission. So where one neuron is silencing the other, or one neuron is activating the other. And when it's called hyperpolarization, where it's inhibiting it, and depolarization when it's activating it. But the neuromodulation, which is happening at synapse at a very specific one neuron to one neuron, you also have the neuromodulatory effects where the neuromodulators are just kind of released from the axon terminals, and one neuron can affect many others. So, which is called the volumetric neurotransmission, and this is point-to-point neurotransmission. But in a real life, you actually basically have both at the synapse, and at the same time, the neurotransmission happening, neuromodulation happening. So the neuromodulatory mainly happens to GPCR, neurotransmissions mainly happen through channels, and through the receptors. And also these neuromodulated GPCR receptors are activating the channels and the neurotransmitter receptor. So altogether, so the neuromodulators are modulating the neurotransmitter receptors. And how do you study that? So there are various ways to, so how do you study the release? And so one way to do is electrophysiology, and then you kind of, so your hypothesis, okay, this happens through NMDA receptors, that is the receptor for, say, glutamate. And so what you do is you put a drug that would block that receptor, and if you then you're recording in PSP, and if that doesn't change, so you don't get the PSP after giving the action potential, exciting the presynaptic neuron, and that would mean that yes, so it was happening through NMDR. So you say, but so the problem with that, not a problem. So the other, there is only one way to measure it, and you're kind of blind about it, but. The recent techniques allows us to actually see them, or what do you mean by that is instead of doing a recording electrophysiologically, we can see changes in fluorescence. The advantage of the kind of detecting changes in fluorescence or imaging is that in electrophysiology, you are kind of blind, because when you express protein, you can tag them with fluorescence, you know which cells you are looking at. And you can limit the expression of this protein to specific kind of cells you are interested in. Electrophysiology, the major disadvantage, you're kind of going blindly because you're not looking at fluorescence. and you kind of and also this one gives you alternate way to look at or study neurotransmission, or when specific neurotransmitter released, how they're released, how much they're released, and if there is a mutation, how does it change this. So there are two complementary ways of studying it, each having its own advantage. So one of the kind of newer ways to study is using ratiometric biosensors, so which are I won't go into the details of it, but the idea basically is that uh, there is one green, and so there is one fluorescent molecule, one side of the GPCR, another side of the GPCR is another molecule. So you are seeing total fluorescence level which you're detecting at certain wavelength. It's kind of quite different from when this GPCR binds to its, say, a neuropeptide ligand. And what happens when the ligand binds to GPCR, it changes the conformation. This green and yellow then comes together, and basically the fluorescent level changes, or the color changes, or it just gets brighter. So by that, so why it's called ratiometric imaging is because you are calculating the fluorescent level here, and then you're making a ratio of it, and that's a readout. And what happens is that using this ratiometric imaging, you can act for specific biosensors, so you can detect, say, substance P versus another neuropeptide when it's released and how it's released. And then so the neurotransmission, so we talked about like what are the things that are happening and what are the major components, but how is it what are the different components of just the neurotransmitter system? So as I said, there are synthesizing enzymes here which are taking the precursors that are coming from the cell body and making them into neurotransmitter. They're vesicle transporters, which means the vesicles are lying around, and something has to introduce the neurotransmitter that being synthesized inside the vesicles. So it's the synaptic vesicles are loading the synaptic vesicles. Then you need reuptake transmitters, because you want to be efficient system, so the neuron are released and taken up, and then once they're taken up, while they're taken up, you need the reuptake transporters. You need degradatory enzymes, so you need molecules that will degrade the extra one, so that a definite amount of vesicles are maintained. Then you have the transmitted gated ion channels, which is the ion channels, the receptors for the neurotransmitters, so the GPCRs, GPCR gated, and secondary messengers. So there are multiple protein components that are here from the moment they receive the precursors. They make it back into synaptic vesicles, and they transmit the information and make sure that there is a signal is kind of transmitted. And so how do we study what are the different neurotransmitters expressed in a part of the cell we're interested in? So one is one way to study that is the electrophysiology technique I was talking about. You're basically recording APSP or IPSPs, and what you're doing is you're blocking the channel and you're sending action potential. And if you're seeing it's blocked, that means, or if you're looking at adding an agonist, or antagonist, then you will see block means that the receptor agonist, you'll get, say, enhanced APSP or IPSP. So that's how we find out. The other way to do that would be to visualize it. So these are all proteins, so we can make antibodies against them, we can stain them, and we can see any cell type of a choice of in any nervous area of the nervous system, what are they expressing, and you can visualize them. So this is which is known as immunohistochemistry. So what it shows here, basically, you take the neuroproteins, the neurotransmitter, you purify them, you inject in a rabbit or a mouse or a rat, they generate antibodies, you purify them, then you conjugate them with the fluorescence, which will be another antibody, but to simply conjugate them with the fluorescence, and then you can, or another, so it's called horseradish peroxidase. But anyway, so you can visualize them in situ, which means that you can look at them on the tissue, and now the cell type of interest is expressing whatever neuropeptide. Also, something that have come up in more recent times, that multiplex in situ hybrid. So it's a very old technique that you look at expression of mRNA. So you are not looking at the protein, then you are asking a cell is expressing mRNA for a particular gene or not. And you can use that mRNA. So using in situ hybridization, you look at the levels. And so you are now imaging the total mRNA in the cell. So this is a multiplex in situ, so you can see multiple colors together. And so what you do here, I mean, the idea is same, that you're looking at the distribution of neurotransmitters. So if you what has happened now is, like, not everybody is an electrophysiologist, not everybody is a can do in situ, but you have complementary ways to look at it. They all have their advantages. Something like electrophysiology even now is very kind of intense, labor intensive, and also it needs a very specific kind of expertise that many people don't have. Whereas here, in situ, the other people can do in situ. So there are different ways to ask these questions. Yeah, and so that's my basically last slide, and so then I'll take questions. Yeah. Yeah, Nila.
So I had a doubt in one of your initial slides. You
Yeah.
mentioned types of synapses, right? One of them was an axoaxonic synapse.
Right.
So how would that synapse contribute to the development of an action potential in the postsynaptic neuron? Because it's not exactly going into the neuron, right? It's going into the axon of the previous neuron. So how does that work?
Right. Uh, so when you say axon, so even though we draw the diagram, right?
Yeah, the third one.
So
Yeah.
let's say here. So this is basically when its axonic synapse is modifying. So this synapse is synapsing onto this cell, right? So it's modifying the signal that this synapse is sending to this cell. Does it make sense? So it's
Okay,
not
so
directly,
it's
so it's not directly communicating with this cell. It's communicating with this cell while this one communicates. So it's changing the kind of, if this say go right, this says say okay, go right by 20 degrees. So it's adds a layer onto the information that passed from this neuron to the other.
okay. 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 So
Yeah.
and one more, in slide 21,
Like,
uh,
okay.
what is
Yeah.
VFPD?
Oh, I have to check. So it's it's something about the ligand that was binding here. I can look it up, but it's
Okay.
not nothing to do with the conceptual thing itself.
Okay. So,
So there is basically a like binding site for this ligand, and that's where it's binds. It's something to do with that. I haven't, sorry. Yeah. So you can just Google that. It's a review. So the Leopold et al.
okay. Okay, sir.
Yeah. Anybody else? I don't see hands. Yeah. Any other?
So there's a question in the chat box.
In the chat box. Yeah, the actisome has a different permeability compared to other parts neuron. Yes. So again, this has to do with the channels that are expressed at the active zone, right? So if I go to that slide, once again. Yeah, so you see that. So it's because of the, so just around the active zone there are this calcium channels, voltage gated, or VGCCs are the voltage gated calcium channels that determines the permeability at the active zones. So that's how the and also the synaptic vesicles are concentrated here, all of them. So this is like a kind of a geographical location that's ripe for fusing of the cell, for fusing for this of these vesicles. And so yeah, so it's determined by a number of factors, and one of them are the expression of voltage-gated calcium channels. Yeah, anybody else?
Sir, is neuromodulation found in all organisms? It's
Yeah.
also a conserved mode of.
Yes, yes. So neuromodulation is conserved through, say, C. elegans, even the smallest of animals to the most evolved of them. So yeah. So a lot of neuromodulation is actually studied in the lobster gut system, because for some reason they are the neurons that controls the gut movement in lobster or feeding has immense number of neuropeptides, and it's very very sophisticated system actually. So a lot of study, what do you know about neuropeptides and how they are changing the computation that's generated or that's done by or executed by a set of neurons is kind of determine the combination in which the neurotransmitters and neuromodulators kind of interacting with each other. A neurotransmitter systems, neuromodulator system, they're acting with each other. Yeah. Anybody else? No. Okay, so we'll stop it here, right? Gene or
tomorrow you are taking class, sir, right?
yeah.
Yeah. Okay. Okay.
All right, bye.
Sir, when will you upload the
And sir,
slides?
could you upload the slide?
Right now I'll upload it.
Yes, sir. You haven't also uploaded the second slide, I guess.
What? You want me to just upload
Sir,
the...
yeah, you also haven't uploaded the second slide. Second lecture.
I did. No? Okay, I'll do it. I thought I did. All right. Thank you.
Okay. Thanks, sir.
Mhm.
Thank you, sir.
Thank you, sir.